Namaskar dear students, welcome you to our channel Bio by Gulab Sir. As a part of series of lectures, we are going to start new chapter, body fluid and its circulation. It can be called as circulatory system. You learn in previous classes about blood, its composition, even heart and its functioning, everything. But still, as it is a chapter in 11th class, you need to have thorough study. So we have this video about body fluid and circulation. So first part of that chapter is blood and its composition. As you know that circulatory system is composed of blood, blood vessels, heart and lymph. So let us start with blood. As we are very much familiar with this word and we very often say about blood. But let you know everything about blood and its composition and function. As you know that blood is very important part to carry oxygen and nutrients to all cells of the body, as well as to collect carbon dioxide and waste material from all the cells of the body. At the same time, it also transfers hormones, enzymes and urea and uric acid produced in our body. That urea and uric acid carried out from all the cells, that ammonia is carried out from all the cells to the liver. In liver, that amino acid ammonia convert into urea, and then urea carry to the kidney to excrete out. At the same time, hormones are produced in glands, endocrine glands. They release into the blood and blood carry that hormones to the target organ. So blood has vital function. No doubt, without oxygen we cannot survive. So we inhale the oxygen, but finally it is being transported to all cells of the body through blood. Now let us know about blood, what its composition is. Blood is a fluid connective tissue. It is a liquid connective tissue. It is liquid because of presence of 90 to 95% of water. And it is tissue because of it has cells and that has specific function. So it is composed of both liquid and solid. So liquid part is called plasma. It is about 55% and it is so called liquid because it has most of the percent is water and it has some dissolved substances that are organic and inorganic substances. So plasma constitutes 55% of the blood. It is composed of organic and inorganic components. Organic components are protein, amino acids, glucose, hormone, urea, uric acid, bilirubin, cholesterol and so on. Very important that which protein? Three types of protein: albumin, globulin and fibrinogen. Albumin help to maintain the osmotic concentration of the blood, that means solute and solvent concentration level. Globulin provide immunity, immunoglobulin, and third fibrinogen that help in blood coagulation, and we are going to learn in detail about blood coagulation under the heading platelets, thrombocytes. Now these components which are present in the blood, they are transported across the body through blood and they have importance, as they have separate importance, that amino acid absorbed into the digestive tract, carry to the cells for the synthesis of protein. Glucose is transported to all cells of the body for the production of energy. Hormones at the target organ to have the action, and urea as a waste material carried out from liver to the kidney to excrete out and so on. Now that inorganic components, as water, most of the part of the blood is water. Carbon dioxide and oxygen, these are the gases which are being transported for the exchange. Salts and minerals, they are suppose carried out from digestive tract to the cells for their functions and importance. So this is what composition of blood as plasma. But what is about that solid part? This was the liquid part. Now what is that solid part? Solid parts are blood cells. They are called as corpuscles because they are free. As other tissue is engaged, it is at the place, it is located at the place. But here these blood cells are continuously moving through blood and therefore they are called as corpuscles. They are also called as formed element because they are produced in bone marrow and from there they release into the blood and then they continuously move. So therefore they are called as formed element. As I hope you got it, the meaning of both the word corpuscles and formed element. Now these corpuscles are of three types. You are aware: RBC, also called erythrocytes, WBC, also called leukocytes, and platelets are also called thrombocytes. No doubt, in detail we have to know about this erythrocyte, leukocyte and thrombocytes. So let us know about them in detail. So it is in continuation. Let's see here RBC, WBC and platelets. I have made this differentiating chart so as to remember easily. So see the first point written about their presence, number of cells present in. So here RBCs are 5 to 6 millions of RBCs per cubic millimeter of blood. WBCs are 6,000 to 8,000 per cubic millimeter of blood, and platelets are 1,50,000 to 3 lakh per cubic millimeter of blood. So you can easily remember in comparison in their number 5 to 6 million, 6,000 to 8,000 and 1,50,000 to 3 lakh per cubic millimeter of the blood. I hope you got it, that first point. Second point, they are unnucleated, biconcave and unnucleated. No doubt, except in llama and camel. Please remember for the NEET that llama and camel, these are the two animals, they have nucleated RBCs. Otherwise, rest of the other mammals, they have unnucleated RBCs. Here, these WBCs are nucleated. Platelets, they are the fragments of megakaryoblast, and since they are fragment, they are irregular and unnucleated. So second point, very clear, that as a differentiating point. Third point, that increase in number of RBC, what happened? No doubt, these are the number, they are confined. If beyond that increases, definitely there will be abnormality, and if decrease in the number, that also cause the abnormality. So what disease or what deformity that we cause because of increasing number of RBC, that is polycythemia, and decrease in number called anemia. The same way, increase in WBC cause leukemia and decreasing WBC cause leukopenia. Here also the same thing, increasing number of platelets called thrombocytosis and decreasing number cause thrombocytopenic purpura. We are not here supposed to see the symptoms of the diseases, but to know about what happens if the number of that cells increase. Okay, that was the next point. Now you all aware that RBCs are red color because of hemoglobin, WBCs are colorless, platelets are also colorless. Next point, form in bone marrow. Yes, all the cells are formed in bone marrow and from the hematopoietic tissue. Bone marrow, no doubt, it is a common stem cells, but the same cells has homopoietic tissue, leukopoietic tissue and megakaryoblast from which these platelets are produced. Once again, erythrocytes, RBCs are produced from hemopoietic tissue. WBCs are produced from leukopoietic tissue and platelets are produced from megakaryoblast. These are very important point to remember as they can be asked in NEET. Next point, the process by which these RBCs are produced, that is called hemopoiesis. Naturally, that for WBC, leukopoiesis and thrombopoiesis. These are the supposed processes by which they are produced. Now, hemopoietic tissue undergo hemopoiesis to produce RBC. Now that they are produced over there and then they release into the blood and then they start the function. But RBC has 4 major steps to be produced. Let us know first there itself how RBC produced in 4 major steps. Here, that young RBCs are nucleated. They are called erythroblast. They convert into normoblast, that is also nucleated. By the time hemoglobin production begins, then they convert to reticulocyte. They are young RBCs here. All the organelles are degenerated. See how RBCs are produced. Mother RBCs are erythroblast as a stem cell in bone marrow. They are nucleated. They convert into normoblast by the process of production of all hemoglobin over there is normoblast. By the time mitochondria, Golgi body, nucleus, endoplasmic reticle, all organelles are present there. But when that concentration of hemoglobin increases, all other organelles get degenerated. Only RBC with cell membrane is present, and that is a young RBC. It is unnucleated RBC. Now, that matured RBC which has no nucleus, that means there is no organelles also. That means they do not use oxygen, but very honestly they carry oxygen to all cells of the body. That is their sacrifice. That themselves, they are not using that oxygen. They are anaerobically respiring. That is the point of NEET. RBC are the cells, although they carry oxygen, but anaerobically respire because they don't have mitochondria. Question can be asked, in our body, a group of cells which anaerobically respire, that is RBC, although that carry oxygen, but see, having oxygen but still doing anaerobic respiration. that is sacrificed, and moreover, they live only for 120 days. By carrying oxygen continuously for 120 days, then they die. After death, they are transferred to the spleen as a graveyard of RBC. Further, from spleen, they transfer to the liver, where they are converted into bilirubin and biliverdin. I hope you got it, that how erythrocytes are formed from erythroblast, normoblasts, reticulocyte and erythrocyte. Come to the previous page, as I told you that their degradation occur in liver, where they convert into bilirubin and biliverdin. RBC degraded, worn out RBC into bilirubin and biliverdin. What are bilirubin and biliverdin? They are green color pigment. When they release into the blood, they combine with red color blood and form the yellow color, and therefore accumulation of these bilirubin and biliverdin in our body, in blood, in eyes, that skin seems to be look yellow color, and therefore we say that we got jaundice. So when liver is ill, by the time liver could not produce bile juice, now liver add that bilirubin and biliverdin into the blood, but since this bile juice is not produced, bilirubin and biliverdin, which supposed to be excrete out, it block and not transferred to the digestive tract, and therefore all that bilirubin and biliverdin which accumulated into the blood gives yellow color, that is called pilia. आपने सुना होगा कि जॉन्डिस को पिलिया भी बोला जाता है, नाखून भी पीले दिखने लगते हैं, आंखें भी पीली दिखने लगती है. It is because of 2-3 types of jaundice. One of the jaundice is obstructive jaundice, and even if liver has got infection, that time also this bilirubin and biliverdin start accumulating in our blood. Now what is the function? That what later happened? This bilirubin which present in the blood is of 2 type bilirubin, urobilin and stercobilin. This urobilin gives yellow color to urine. Our urine is yellow color is because of this urochrome produced from bilirubin, and stercobilin is another form of bilirubin that gives yellow color to our tools, that is fecal matter, our excreta. Do you know that when doctor is supposed to diagnose you for jaundice, they ask for your fecal matter. Is it dark color or yellow color, or they ask for the fecal matter, they collect and observe that if it is dark color, definitely you got the jaundice. Why? Because yellow color bilirubin is not coming into the excreta, that means it is obstructed into the bile duct, and therefore that you have dark color excreta, and that indicate you got the jaundice. At the same time, if start appearing yellow color eyes or skin, it confirmed to be jaundice. I hope you got it, that what happened with RBCs when they are dark. It is what final, that is the destiny of RBC. After 120 days, they are being degraded by liver. Liver does the very fantastic function of that removal of that RBCs from our body, dead RBCs from our body. I hope you got it. Now at the same time, then what is the lifespan of WBC? RBC has got 120 days lifespan. Then what is the lifespan of WBC? Here it is clearly written, they get degraded when exposed to the pathogen or antigen or memory cell, that they remain as a memory cell for many days, but otherwise within 4 to 5 days or 6 to 7 days they get degraded. So their lifespan is very short, but some of the cells, they form the memory cell. When they expose to the antigen, they convert into memory cells and they live for years. Therefore, some of the diseases we get once, but second time we do not get that disease because of that memory cells which fight against that pathogen if it is inhaled in our body, and that's what you know very well. What are about platelets? Now platelets are so short-lived, but when they rupture at the site of injury, that we get that injury, while injury we start bleeding, that part start bleeding. Now these platelets help to coagulate the blood. That process we are going to learn in detail. I hope you got it, the difference between RBC, WBC and platelet in detail. Now let you know about again detailed information about WBC. We have discussion about RBC. Now let us discuss about WBC. WBC are white blood corpuscles. They are soldier of our body. They provide immunity to our body. They have very vital role, but still in detail about their components, that they are divided into 2 types, granulocytes and agranulocytes. They are so called granulocytes because see that they have granular cytoplasm, and here they are called as agranulocyte because cytoplasm is spread. plain. There is no granular cytoplasm. Moreover, they are differentiated on the basis of different shape of nucleus. Now see, granulocytes are of three types on the basis of shape of the nucleus and their function. Neutrophil, acidophil, basophil. Acidophil also known as eosinophil. Let you know that one. See, the nucleus in neutrophil is multilobed, in acidophil it is bilobed, and in basophil it is S-shaped. Now what are the function? That neutrophil is phagocytic in nature, it directly engulf the foreign material and pathogen and destroy them. That's very important function. It is polymorphic nucleus. Since multilobed nucleus, this is called as polymorphic nucleus. Acidophil has very important function that they register infection as well as they help to combat with allergy. So remember this point, resist infection as well as produce antitoxins against allergen. Allergen मतलब those who produce allergy, जिससे हमको एलर्जी होती है उनको allergens कहा जाता है, और उसके खिलाफ कौन काम करता है? तो एसिडोफिल काम करते हैं. समझे? Basophil, they are special cells, they produce histamine, heparin and serotonin. See, they are also known as mast cell of blood. Why? Because they produce histamine. What is the function of histamine? It produces inflammation. Serotonin is very important hormone which gives sense of happiness, and heparin is an anticoagulant. So WBC, basophil has these three main functions. Okay. Lymphocytes, they are as equal as size of this granulocyte, but they have very large size nucleus as compared to the area of that cell, and therefore it can be recognized as it has a very large size nucleus. This has again importance, very important. Lymphocytes are of two types. They are B cell and T cell. Those lymphocytes which get matured in the bone marrow, no doubt produce in bone marrow, but get matured also in bone marrow, are called B cell, and those which get matured in thymus gland, they are called as T cell. These B cells, they produce antibodies, specific antibodies against specific antigen to destroy them. So very vital function. T cells which get matured in thymus gland, that they are of three types: helper T cell, killer T cell and suppressor T cell. What is the function of helper T cell? They help B cell to produce the antibody. Killer T cell directly kill the pathogen, and suppressor T cell suppresses our immunity going against us. That means suppresses autoimmunity. Very important. Our immunity may destroy our body, and we have some autoimmune disorder also. In that disorder, our immunity goes against us, like myasthenia gravis, rheumatoid arthritis, even in AIDS also. Our immunity start eating us. Therefore, when AIDS related complex occur at the age of, after having that AIDS for 10 to 12 years, that our body suddenly weight start decreasing. That means our immunity start eating our own cell. That is what autoimmune disorder. I hope you got it. These two cells, bone marrow and thymus gland, B cell producing bone marrow, T cell get mature in thymus gland. Therefore, these two structures, bone marrow and thymus gland, are called as primary lymphoid organs, and so on. So the types of this B cell and T cell. I hope you got about lymphocytes. Monocyte, they are the largest blood cell with kidney-shaped nucleus. They are also phagocytic in nature, that they engulf the foreign material, pathogen and destroy them. So lymphocyte, this monocyte and neutrophil, they are phagocytic in nature. I hope you got this classification of blood cell in detail. Also, you have to remember how much percentage of they occupy in that blood as a blood cells. That neutrophil around 60 to 65%. The highest number of blood cells, that is WBCs, are neutrophil. Then 20 to 25% lymphocytes. That you remember, please. 20 to 25% lymphocytes. Monocytes are 6 to 8%. Who are the highest? Neutrophil. Who are the least? Let you know, basophil. So highest in number neutrophil, less in number basophil. Question can be asked. One of the following is a correct sequence of decreasing order of their size. This can be arranged in different order, but you have to select decreasing order. That means first will come neutrophil, last will come basophil. I hope you got it. That sense. Question which can be asked in NEET. Okay. So these are the suppose some of the points which we have discussed about different types of WBCs. Now let us know about platelets. Last, different types of cell. The platelets are also known as thrombocytes. At the site of injury, their function is very important. You know that their presence is 1,50,000 to 3 lakh. If their number decreases, in which disease? you are aware about that their number decreases? Dengue. When we get dengue, suddenly platelets decreases. And doctor says platelets come to the 1 lakh, 1,50,000 or very less number. So that is the sign of dengue. But what is the function of platelet in our body? At the site of injury, when we get cut or wound, blood start oozing out. Now that bleeding of blood has to be stopped. At the site of that injury, at the same time when blood oozing out, platelets get injured and they produce thromboplastin. That what they release, the thromboplastin is also known as thrombokinase. This is very important. It catalyzes the process of conversion of inactive prothrombin. It is a plasma protein. It is so insoluble form in plasma. Inactive form, prothrombin, convert into its active form, thrombin. Who convert it? Thromboplastin. Ultimately, this thrombin activates fibrinogen into fibrin. That means it converts soluble fibrinogen protein from plasma into insoluble form, that is mesh, that is fiber-like structure. This fiber-like structure, when produced at the site of injury, they form the mesh, that means net. And that dense net attack dead cells of the blood, and thereby it begins to get dry, and that form the clot, and thereby when blood oozes out, it suddenly stops. After 5 to 10 minutes, within 5 to 10 minutes, or even within 2 to 3 minutes, that blood get coagulated by this process and stop the oozing of blood, that we call clotting of blood, also known as blood coagulation. So see the role of platelets. It stops our blood oozing at the site of injury. See the role of this enzyme thrombokinase. Remember this. Also, other 13 factors that they play a role in blood coagulation. Out of that, these are the 4 factors. Please remember, fibrinogen is the first factor, prothrombin is the second factor, thromboplastin is the third factor, and calcium, which acts as a cofactor with this thrombokinase enzyme to convert prothrombin into thrombin, is a 4th factor, and other factors including 13 factors that play vital role that we are going to learn in genetics. I hope you got it this in detail about blood and its composition. Even if you are in doubt, you can ask in comment section. I hope you like this video. Also, I will appeal you to share this video with your friend and subscribe our channel if you are new. Also, I request you to press bell icon to receive your notification as and when new videos are uploaded. Thank you. Enjoy the learning.